Hi everyone. This week in lab we are doing lab number 5 in your lab manual, which is environmental stress and diversity. So this particular lab, if we were on campus, we would be going to Harper Lake and collecting buckets of water and then bringing it back into the lab, and we would be looking for the little things that live inside the water. So if you remember way back to our very first lab when we did those salt tests and you got to pick either amphipods or Daphnia, and you got to see how much salt that they can tolerate. So in this particular lab, we're going to be looking at a much bigger variety of those little things that live in the water. So we call these benthic macroinvertebrates, and they're called benthic because they sort of live near the bottom of the pond, and then they'll swim around, and that's when we catch them. And then they're macroinvertebrates because we can see them without the use of a microscope. You can certainly use a microscope to get a better look at them, but you don't have to have one. So we don't have an opportunity to go out and actually sample the water. But what I did want to show you, so I'm going to go ahead and go here into our weekly folder. Excuse me, go into lab materials. What I did want to show you is a link that I have provided for you. It's kind of like a virtual macro collection lab. So when you follow it, it takes you here to the benthic macroinvertebrate portal. You can read some information about stream sampling. You can take a look at what these things actually would look like under a microscope. So they're gonna sort of show you some different things here, what these would look like next to, again, with the size of a penny, just for size comparison. So you would get to see a whole lot of different things, again, if we were in lab together and if we were actually able to see what these things look like. So it also tells you what benthic macroinvertebrates are. There's even the ability to do like a virtual stream sampling. So when you click on this link, you can pick a site to sample. And when you pick the site to sample, so you will actually grab a net here. And so we need to, oops, I'm sorry, let's go back. So this is for the macros. Oh, and you grab your net, I'm sorry. You grab your net and then you put it in the water, pretend that's what you do, and then you sort through all of the leaf litter and you find different things that you can find. It's like, oh, let's see what you found. So we're going to put it in our little tray here, and you're going to try to figure out what it is. And again, this is much more complex necessarily than what we'd be doing. We have some picture guides and things to help you figure out versus using a key. But I did want to let you know that there is an option here if it is something that you're really interested in and you want to learn more about it. This is a really great site to look at. So what we're going to be doing is I am, just like in previous weeks, I have given you the data for this week's lab. It is in a spreadsheet again, so this spreadsheet has two types of data in it. It has data for the macro, and also as part of this lab, in addition to looking at the macroinvertebrate data, we're also going to be looking at algae. So in lab, normally you would come in, you'd get your samples, you would start separating your species out, and you would try to identify them. I've done this for you. I've given you a whole list of what invertebrate species we collected in our sample here, and also what algal species we have collected. So we have, again, invertebrates, animals, and algae, which are kind of related to plants. So that's kind of part one of this, is you're going to look at that data. And then part two is you are going to fill out table 5.1 in your lab. Okay, so here's table 5.1. And what table 5.1 wants you to do is it's asking you to put these species into different categories. So you'll see that there's a group 1, a group 2, a group 3, and a group 4 on here. What those groupings have to do with is they have to do with pollution tolerance. So some macros are very, very sensitive to pollution. So if we have too many nitrates or too many phosphates, or we have other types of contaminants in the water, they're not going to be able to survive, so you won't find them there. And then there are other macros that are much less picky. They'll live just about anywhere they can find water. So they're very pollution tolerant. So the next thing that you're going to do is, with the data, you're going to open this PDF, and the PDF has all of the species in here, and they're grouped according to whether they're group 1, 2, 3, or 4, which again is clean all the way down to species that are very pollutant tolerant. So you'll go back to that data sheet that I gave you, and what you're going to do is you're going to find out where these species are on one of these little picture guides. So for example, if I'm going back to my data here, let's look for the water penny beetle larvae. Okay, so let's just do an example and let's look for the water penny beetle larvae. So let's see, I'm going to start in group 1, and I don't see anything that looks like a water penny, so I'm going to move up here to group 2, and I'm looking through, and oh, here it is. Here's my water penny beetle larvae. Great, I identified it. It's a group 2 species. So I'm going to come back to my lab, and in group 2, I'm going to write down water penny beetle larvae. Okay, so you will do that for all of the species of both, start with invertebrates first, of all of the invertebrates. So you're going to sort them all into these different groupings, and then you're going to come up with a total. Okay, so at the bottom there's a total for each number, how many 1, 2, 3s, and 4s that we have. Okay, once you've done that, you're going to go to the next page and you're going to figure out the pollution tolerance index. Okay, we use P to describe that. So pollution tolerance is really easy to figure out here. So what you'll do is, here, let me go back to the lab. What you'll do is you're going to take the number of species in each group and write that number here. Okay, then you're going to multiply by whatever number is in this column, and then you're going to put the new number here, add it up, and get a total. Okay, so a total big T. You take, to figure out the pollution tolerance index, you take big T, which is the sum of all these numbers, divided by little t, which was the sum of all these numbers, okay? And it's going to give you a value. And that value is going to tell you a little bit about the water quality of our lakes, according to the macroinvertebrates that live there. So depending on what the number is, depends on what the quality is. And in this case, the lower the number, the better. The lower the number means we have more group 1 species, which group 1 species are the least pollution tolerant. So that's kind of what we're after here. So then you'll find out which categories here. Okay, you're going to write the number down, and then you're also going to write down the qualitative, which is this part, which is excellent, good, fair, okay, or poor. All right, so that's part 1. You're going to do that with the macros. Part 2, you're going to turn the page and do the exact same thing we just did, but you're going to do it with the algae species instead. So I also have all of the algae pictures for you. So here they are, and please note that the group numbers are a little less obvious on this one. Look in the top left-hand corner to find the group numbers. But once again, group 1 are the clean algal species. Group 2 are moderate. Group 3 are more tolerant to pollution, and group 4 are the most tolerant to pollution. Okay, so once again, you'll do exactly what you did before. You're going to go to the spreadsheet. You're going to find the algae. So let's do Chlamydomonas. Okay, I've always liked Chlamydomonas. So if you go back to our pages here, we're going to look for Chlamydomonas. And I think the algae are a little bit harder than the invertebrates, to be honest with you, because we have a lot more examples of algae on each sheet than we do have with the macros. But at the same time, the algae are kind of nice because they're all in color. Although algae are really only 2 color. They're either green or they're brown, to be honest with you. So in any case, I know where Chlamydomonas is. Chlamydomonas happens to be a polluted water algal species, so it's right here. It's this thing that kind of looks like a circle with an eye spot and 2 tails. Little alien looking like. So that's Chlamydomonas. So then on my sheet, I would go to group 4 and I would write Chlamydomonas, okay? I'll do the same thing that I did in the previous. I'll add the total up of all of the columns. Okay. Then I go down here and I do the pollution tolerance index again. So writing in the number of each of the species in each group, multiplying across, okay? Dividing big T by, whoops, this way, little t, and then once again coming up with our pollution index, okay? So that does it for the lab work. Please keep in mind that there are lab questions for this week. So for the lab questions, I'm having you do 3, 4, 5, and 6. So as before, hold on to all of that. A lot is going to change probably between now and the end of April, so I will sort of wait until I have more information before I figure out how I'll have you all submit these pages to me. But that is it as far as lab goes this week. Don't forget that the lab quiz covers the soil lab from last week, right? The questions that you did on the soil lab, and then also one question from this week's lab. So please don't hesitate to ask if you have any questions.